Hi everyone, my name is Clement. Today I'll be presenting findings from my honours project titled Contrasting Drivers of Snapper Growth Along the East and West Coast of Australia. Humans are drastically altering marine ecosystems. Fishing increases mortality rates and alters density-dependent processes through the selective removal of individuals, while climate change can expose organisms to stressful environments. Despite being underpinned by different mechanisms, fishing and climate change can lead to the expression of faster life histories characterized by shrinking adult body sizes, changes in juvenile growth rates to attain earlier maturation, and lead to reductions in expected lifespans. However, despite evidence suggesting that fishing and climate change can produce similar life history trends, most studies examine these factors in isolation. Potential interactions have only been considered in the last decade. For example, size-selective fishing can alter the timing of key reproductive events, leading to potential mismatches between the spawning period and timing of food availability. Fishing can also increase the vulnerability of fish to climate change by selecting against certain phenotypes with the adaptive capacity to respond to warming. More importantly, fishing and climate change reduces population diversity and can lead to greater synchrony among fish populations. This increase in synchrony can enhance the vulnerability of fish populations to stochastic environmental events, such as heat waves, as declining populations are less likely to receive demographic rescue from nearby populations. Such changes can have broader implications for individual fitness, population dynamics, as well as fishery productivity. Understanding the degree to which fishing activity interacts with temperature is crucial, as fisheries provide food for billions of people every year. Growth is a fundamental biological process that describes the change in individual body size over time. It is an ideal candidate to study the effects of fishing and climate change, as growth is sensitive to intrinsic or within individual differences, as well as extrinsic or environmental factors. And while growth is an individual trait, it underpins a suite of population-level matrices, such as recruitment variability and stock biomass, both of which are crucial for our understanding of fish population dynamics. Many fish routinely deposit material into their otoliths, forming alternating opaque and translucent growth rings. By measuring the widths of these growth rings, we are able to reconstruct the growth histories of individual fish. The aim of my honours study is to determine the relative importance of temperature and fishing activity on the average annual growth of snapper along the east and west coast of Australia. Snapper, or Chrysophrys auratus, is a long-lived fish with an extensive distribution from Western Australia throughout southern mainland waters to Queensland. Snapper's extensive range mean that populations are exposed to a wide range of environmental conditions as well as fishing activity. Furthermore, the southward flowing warm ocean currents along the east and west coast provide a unique opportunity to study how the growth of this commercially and recreationally important fish varies along an environmental gradient, as well as to check the generalization of my findings. Snapper is managed as 4 separate bioregions throughout my study area. We have the Queensland and New South Wales bioregions on the east coast, the Gascoyne and the west coast bioregions on the west. I selected 15 sites along both coasts, each site corresponding to fishing zones in each bioregion. Otoliths were then selected based on their capture year, as well as estimated age, to ensure good coverage of the age classes throughout the reconstructed time series. But because my honours project was being conducted during a pandemic, I had to make some contingency plans to get around border closures. I visited the New South Wales Fishery Lab in March 2020 before lockdown restrictions were imposed to image their otolith collection. The Fishery Lab in Western Australia sent 20 kilograms of otoliths to my apartment in Melbourne, and Queensland Fisheries sent a hard drive containing thousands of otolith images. As you can see from the image on the right, I ended up doing all my increment measurements at home. Next, I examined a suite of predictor variables. I had 3 temperature variables: average annual sea surface temperature, average temperature from spring and summer, as well as the lagged annual sea surface temperature. These variables allow me to explore if average growth is related to current temperature conditions, conditions during the warmest months of the year, or if it's from the conditions from the previous year. I also examined 2 fishery variables, namely catch per unit effort, which is an indicator of relative abundance of fish in a bioregion, as well as total catch, indirect proxy for fishing selectivity. I decompose each predictor variable into their among and within site components to explore potential underlying spatial and temporal trends. Let me explain this with an example. Here, we have a graph illustrating the temperature of 2 sites over time. We have temperature on the y-axis and time in years on the x-axis. The red line represents a warmer site, while the blue line represents a cool site. I calculated the long-term average temperature of each site, which represents the spatial component of temperature along a latitudinal gradient. Next, between the temperature of a given year and that of the long-term average temperature to determine if it was a warmer or cooler year than average, as represented by the vertical lines on the graph. A series of mixed effects models were then used to disentangle the relative importance of intrinsic or within individual differences, as well as the extrinsic and environmental drivers of average snapper growth. First, I fitted a series of only models, perform model selection on these models to identify the predictor variables that best explain snapper growth. Next, I fitted interactions between the best temperature and fishing variable to explore if there were any potential interactions on growth. Moving on to my results, I counted a total of 37,481 rings and reconstructed the growth histories of 5,769 fish with biochronologies dating back to 1965. Here, we found that average snapper growth varied both within and between sites, along both the east and the west coast, and over time. Here is a graph of relative annual growth of snapper between sites and over time. We have relative annual growth of snapper on the y-axis and time in years on the x-axis. Each colored line represents the average growth of all snapper. And here, we can see that relative growth varied considerably between sites and over time, and between both coasts as well. Moving on to my intrinsic models, age was the best intrinsic predictor of growth, with growth decreasing with increasing age. There were age-specific differences in growth rates between the east and the west coast, with faster growth rates occurring on the west coast compared to the east. Next, I found that average snapper growth was best explained by the additive effects of average annual sea surface temperature, as well as within bioregion CPUE, or the proxy for abundance, along both coasts. However, the relative importance of each predictor variable varied considerably between coasts. I'll first compare the results, the relationship between temperature and average snapper growth along both coasts before proceeding the results between density and growth. I found a positive relationship between average snapper growth and among site sea surface temperature on the east coast. Graph of the relationship between average growth, which is on the y-axis, as well as site temperature, which is on the x-axis. Here, we can see that average growth of snapper is higher at warmer sites and lower at cooler sites and this result is consistent with the temperature size rule, which states that higher temperatures increase growth rates, but decrease the body size of individuals. Empirical observations of fish size in the field. Temperature can affect an organism's growth rate through direct effects of temperature on biochemical reactions in the body, which are key to energetic processes such as metabolism. Another plausible explanation is due to trade-offs among life history traits. Life history theory is based on the idea that combinations of traits are constrained by the trade-offs among traits. Selection may favor individuals with slower growth rates at high latitudes or cooler sites to attain larger size at ages. Recent work by Flickek et al. 2020 showed that snapper growth proxied by body weight decreased during winter. It is possible that starvation mortality during winter may select for larger body sizes, as larger fish tend to have higher energy reserves and are better able to withstand cold temperatures compared to smaller fish, thus resulting in the slower growth rates. Moving on to the West Coast, I found a curvilinear relationship between average growth, which is once again on the y-axis, as well as within site average annual sea surface temperature variation on the x-axis. Here, we see that average snapper growth was optimized at about 0.5 degrees warmer than the average site temperature. Warming is thus likely to benefit growth up to a certain point, after which growth starts to decline. It is likely that fish from the West Coast are living close to their terminal. Interestingly, the best extrinsic predictor of growth along the West Coast was a within-site variable, and this suggests that fish growth is driven by temperature fluctuations in a given site, suggesting that fish are adapted to local conditions. Indeed, snapper has been shown to show strong site fidelity with small home ranges of about 20 kilometers, while previous studies have shown that the West Coast is made up of a single genetic stock. Future studies can compare genetic markers of different populations along the coastline to determine if populations are locally adapted to the various sites. Moving on to my density-dependent results, I found contrasting relationships between the East and the West Coast respectively. I'll start by describing results from the East before talking about the West. Here, I found a negative relationship between average growth, which is again on the y-axis, and abundance on the x-axis along the East Coast. We have average growth was reduced at high abundances and it increased at lower abundances. However, there was a positive and contrasting relationship along the West Coast, where we see a positive relationship with growth increasing with abundance. The likely explanation for the negative relationship along the East Coast is that fishing may be releasing populations from compensatory density-dependent processes, such as intra-specific competition for resources. The selective removal of the larger and more dominant individuals in the population may free up resources for smaller, shyer, and less dominant individuals to utilize, thus resulting in an increase in the average growth rates of these individuals. What about the West Coast, where there was a positive relationship between abundance and growth? One plausible explanation is that snapper growth is dependent on the cohort strength or the number of individuals hatched in a particular spawning season. Indeed, previous work by Hammer and Jenkins found that snapper expresses strong inter-annual variability in recruitment. Weak recruitment years associated with availability or poor environmental conditions may lead to decreases in larval growth rates and survival based on the growth mortality hypothesis. Differences in cohort strength may consequently affect snapper growth through density-dependent processes, such as intraspecific competition, as well as carryover effects from the larval to the adult phase. And while I did not explore any cohort-specific growth patterns in my study, I intend to conduct further analysis to explore potential carryover effects. However, I should point out that I did not fit in any life stage interactions between my predictor variables and growth. We know that temperature can have varying effects on fish from different life stages, and fishing is often targeted at larger and thus older fish. I intend to fit these interactions for my future paper to explore if there are any potential three-way interactions. Another limitation is that fishing activity were based on statewide estimates on a bioregion level. However, abundance varies at much finer spatial scales. Finally, I did not include any recreational fishing data into my analysis. Recreational fishing makes up a substantial portion of total catches, and the lack of such data may influence our interpretation of growth data. In conclusion, I found that average snapper growth was best explained by the additive effects of average annual sea surface temperature and abundance. However, the growth responses varied between the east and the west coast, and this highlights the complexity of studying temperature-fishing interactions on growth. The next step for my work is to explore potential interactions between life stages, temperature, and fishing activity. Thank you for your attention, and I would like to thank the following people for uh, their kind collaborations. Thank you.